Welcome to the Chemistry, Biology, and Math Revision Hub. This video is part of the Biology Topics Revision series. Today we are looking at the components of blood. So let's begin. So the components of blood include, of course, blood plasma. As we can see here, there is plasma, and plasma contains ions. Ions could be calcium, they could be sodium, they could be potassium, and all. And then they include water. Then we have proteins. Proteins, this could be blood proteins or proteins that are like maybe enzymes and so on. So here we can see there are nutrients like glucose and so on, and wastes like urea. Then finally we have gases that could be carbon dioxide, as well as other gases that are present in there. The other part is the white blood cells or blood cells, which include the white blood cells as well as the red blood cells. Among the white blood cells, we have the lymphocytes as well as the phagocytes. And then we have the other types of blood cells called the platelets. And finally, the red blood cells. So blood cells include this one here, the white blood cells, the platelets, as well as the red blood cells. And under subclassification of the white blood cells, we have the lymphocytes and the phagocytes. So here I'll begin with plasma. Plasma, which is the liquid part of blood, is composed mainly of water, but it carries cells around the body. Plasma also carries nutrients, it carries hormones, it carries carbon dioxide as well as urea across the body. Plasma also distributes heat around the body. So I'll continue to the next page. So the red blood cells sometimes are called erythrocytes. These are biconcave in shape. As you can see, the shape here is biconcave, or they can be disc-like cells with no nucleus. They have no nucleus so that there is a large surface area for the transportation of oxygen. So the lack of nucleus, so here they say lack of a nucleus enables them to have a large surface area to volume ratio, to have a lot of hemoglobin in order to carry more oxygen. So that is a good adaptation in enabling the red blood cells to carry out their function. Also, they have a thin shape to provide a shorter distance for diffusion. Everybody, you have to remember, if you're talking about diffusion, think about surface area to volume ratio, and also you have to talk about the distance the substances have to diffuse. If it's shorter, then the rate of diffusion will be faster or be higher. Another thing we know is that there are many millions of these red blood cells per millimeter cubed of blood, and they also transport oxygen around the body. Red blood cells contain hemoglobin that are to load oxygen in the lungs, and then to unload or offload oxygen when the blood reaches the tissues. Red blood cells are made in the bone marrow, and they live for about 100 days, and they are destroyed in the spleen. So that is all to know about red blood cells. Now, still talking about red blood cells, I'd like to include hemoglobin in this topic. So hemoglobin is an iron-containing protein found in the red blood cells that combines with oxygen to carry it around the body. If this is hemoglobin, the iron-containing protein, if it combines with oxygen, it's going to form oxyhemoglobin. This occurs in the lungs, as you can see, this direction. Formation of oxyhemoglobin occurs in the lungs, and then the backward reaction when oxyhemoglobin separates, releasing oxygen and hemoglobin, it occurs in the tissues when the red blood cells are releasing oxygen to the tissues. So that's it. Let's continue to the next part. The second type of blood cells we're going to talk about are the white blood cells, and these include two major types. Actually, the way we teach white blood cells depends on the level of the student. So for lower levels, I would say middle school, we just say they are phagocytes. We say phagocytes, and then we can say they are lymphocytes. Let me write it here. However, when you go to advanced level, these lymphocytes can be divided into other cells like the B cells, the T cells, and so on. So this one here in this video, I'm just subdividing them into the simplest possible part, which is phagocytes and lymphocytes, and that will be it. So I said white blood cells are divided into lymphocytes and phagocytes, and they protect our bodies against invasion from pathogens. I would say from disease or pathogens. Usually pathogens are disease-causing organisms. So the first part is phagocytes. They change their shape and produce extensions of their cytoplasm, as you can see here. This is an extension that is created. So we see the pseudopodia surround and engulf the microorganism into a vacuole. So here they surround it, as you can see here, and then it's engulfed into a vacuole. Enzymes are secreted inside the vacuole. These enzymes will break the microorganism within we call it the phagosome. And then phagocytosis also means cell eating. So this process here you see is like, it looks like the white blood cell or the phagocyte is engulfing the microorganism. In this case here, we wrote a food particle, but that can also be a microorganism. This demonstration here is about phagocytosis. So a microorganism could be engulfed into an enclosure, which is a vacuole, and the enzymes are going to be released into that space. Here, the enzymes will digest or break apart, as you can see here, the microorganism, I'll say the pathogen. It's going to be broken down, and then that's it. It will be destroyed. Let's go to the next page. Another form of white blood cells includes the lymphocytes. Remember, we have talked about the phagocytes. Lymphocytes are different because they do produce chemicals called antibodies. Antibodies are soluble proteins that can be present in blood but move into specific areas where where their purpose is required. So here we say that these proteins pass through the plasma, which is the liquid part of blood, and pathogens like bacteria and viruses that have antigens. If this one here is a pathogen, pathogens do have molecules on their surfaces. Those molecules, we call them the antigens. And so on top of the antigen, that's where the antibody will bind. So antibodies are complementary to the antigen found on the surface of the pathogen. So these antibodies stick onto the surfaces of the antigens and cause the destruction of those antigens in various ways. We will look at the ways. So when we talk about the antibodies, these are, of course, we say they are proteins in nature, and they stick onto the surface of the pathogens or the antigen. They bind to the antigen found on the surface of the pathogen, and they cause the pathogens to stick together. When they do stick together, phagocytosis can occur rapidly, meaning if this is let's say here, like that, if that is the phagocyte, it can engulf so many pathogens because they will be clumped up together due to the action of the antibodies. So instead of the phagocyte consuming one pathogen at a time, it can consume many pathogens within one phagocytotic move. The next part, how antibodies can destroy pathogens. They can cause the labeling, or they mark the pathogen. We call this labeling of the pathogen, so that phagocytes can recognize them and destroy them. So this causes a rapid destruction of the pathogens. The other part is they can cause some pathogens like bacteria to burst open. When they burst open, it means they are going to be destroyed, so they cannot infect newer cells. And lastly, they can neutralize the toxins or poisons produced by the pathogens. Some pathogens like bacteria do produce toxins that can infect other cells or cause they can weaken the burst or cause disease or cause side effects. So to prevent this, some of these toxins that are released can be neutralized by the antibodies, and that prevents further symptoms of the infection from occurring. Moving on. Lastly, I want us to talk about immunity. Like we know, immunity is in two forms. There is what we call passive, and there is what we call active. Active immunity involves formation of memory cells. Here, memory cells will be produced. And we know that antibodies are going to be produced as well. Antibodies produced. In passive immunity, here there is no need for formation of memory cells. Antibodies are not necessarily produced, but they could be donated. So let's see examples of passive immunity. We can see like when somebody receives antibodies, receiving antibodies from a donor, that is not active immunity, or breastfeeding from a mother to the child. When the breast milk transfers antibodies to the baby, that is passive immunity. However, active immunity can involve two forms. One is when the person is really infected by the disease or the pathogen, and then the symptoms are observed. The immune system works, produces memory cells as well as antibodies. That is active immunity. The other one is when there is vaccination. If vaccination occurs and the immune system works harder, leading to the formation of memory cells, that is also active immunity. So here it involves memory cells. So here, lymphocytes do not kill microorganisms straight away because they have to develop into those ones that do not kill the microorganisms straight away will develop into memory cells, and these memory cells will remain in the blood or in the body for many years. If there is ever reinfection that occurs, these memory cells will be activated and they will produce antibodies within a shorter period of time, and larger quantities of antibodies will be produced, and this will help deal with the pathogen to prevent the infected person from experiencing the symptoms of disease. And so we can see here that this is secondary immune response. Usually, if somebody has been exposed to the infection previously, secondary immune response is going to be faster in comparison to primary immune response. Next part, let's talk about vaccination. Vaccination creates immunity to a disease-causing microorganism or organism without somebody experiencing the real disease. So if an agent that carries the same antigen, remember, these antibodies respond to a specific antigen, and when they do respond, memory cells are going to be created. So in this case, if somebody is vaccinated with the same antigen or with some agent containing the same antigen as that found on the surface of the pathogen, the body is going to be tricked into thinking that the person has been infected with a real pathogen. So the lymphocyte will recognize the antigen and they will produce antibodies. When antibodies are produced, also some lymphocytes that are not involved in the production of antibodies will form memory cells that remain in the body. If there is reinfection, secondary immune response takes place, and this is going to be faster. Antibodies will be produced faster, and the infection will not be felt. So this graph here shows us the difference between primary immune response and secondary immune response. We can see for primary immune response, it actually takes a longer period of time. You can see the maximum is obtained here, but it takes a longer period of time for obtaining the maximum concentration of antibodies. However, even the maximum is not high enough as what we see in the secondary immune response. For secondary immune response, when infection occurs here, we can see it takes a shorter period of time to have a higher concentration of antibodies, which is more than 2 times the concentration of antibodies that was reached with primary immune response. So we can see secondary immune response is going to be faster because antibodies are produced within a shorter period of time and in high concentration. Lastly, let's look at the common vaccinations. Some vaccinations can be given when a weakened strain of an actual microorganism is given. This includes in the case of polio, the cases of tuberculosis, as well as measles. Sometimes they can give you a dead microorganism like typhoid and whooping cough, or you can be given a modified toxin of a bacteria like in tetanus as well as diphtheria. And sometimes people can just get the antigen, like in the cases of the influenza. The third type of blood cells are the platelets. Platelets are fragments of cells that help repair damaged tissues in case of cuts. We'll begin by looking at how platelets work. If there is exposure of the open skin to air, this stimulates the platelets and the damaged tissues to produce a chemical. This chemical here makes the soluble protein, fibrinogen, to be converted into the insoluble fibers of a protein, fibrin. The explanation here is about middle school level, not high school biology. High school biology, A-level biology, and other high levels of biology, you have to talk about other proteins that occur or participate in the clotting of blood before fibrinogen. You will talk about this chemical here. We'll talk about it in the next video that I'll make. I'll talk about the advanced level of blood clotting involving other advanced proteins. But for now, let's just talk about just fibrinogen being converted into the insoluble protein fibrin. The fibrin forms a mesh or a network like this one here. You can see that mesh-like structure around the wound or around the open area, and the red blood cells will get trapped into that area. Again, you can see here, you can see those red blood cells, that one, and that one. They are trapped by the fibrin around that area, and when this occurs, it forms a clot that prevents further loss of blood, and it also prevents the entrance of other pathogens that could infect or cause the problems to the damaged area. The clot develops into a scab that protects the damaged tissue. If you look at this part here, there is a damage, and then the protein fibrin forms a network that traps blood cells, including red blood cells and other things that lead to formation of a dried scab-like structure that prevents excessive blood loss. This brings us to the end of this topic. Thank you for being with us. And please do not forget to subscribe to our channel. I'll see you in our next video. Bye-bye.